Hello, my name is Dr. Ernest Ricks, and today we'll be discussing non-coding RNAs in the human genome. This is a fascinating topic. Now, you've probably heard of the central dogma, right? I know you've heard of the central dogma because we've talked about it so many times, right? But let's go over it one more time. DNA gets transcribed into what? RNA. RNA gets translated into protein, right? And I pause just so you can say it with me basically. DNA gets transcribed into RNA gets translated into. All right, awesome. But here's something interesting. Not all RNA gets translated into protein. Some RNAs do other amazing things, and that's exactly what we're going to talk about in this lecture. So, of all the RNA that's created, less than 3% of it gets translated into protein. Even though that's the majority of what gets talked about is the proteins and the genes that are being moved on into protein. That's what we talk about the most, big functions. That's such major functions. However, because proteins have so many functions, they're enzymes, right? They're involved in so many different things. However, these RNAs have function as well. They play a role in gene expression regulation. There are multiple types of these non-coding RNAs. There are tRNAs, and there are rRNAs, and these are some of the ones that you've probably heard about the most. So the tRNAs and the rRNAs are the ones you've probably heard about the most because they've been involved in processes like translation, right? However, there are other ones that are popular as well, popular in science. And I don't know if you've heard of them or not, but there are microRNAs and small interfering RNAs, and they are directly involved in gene expression regulation. So they're part of those small non-coding RNAs. And then there are long non-coding RNAs that play a role in gene expression regulation as well. We'll start off talking about the most popular ones, microRNAs and siRNAs. So these two subtypes of non-coding RNAs are heavily involved in post-transcriptional regulation of gene expression, meaning they're active after transcription has occurred. They're involved in a process called RNA interference, or RNAi, where RNA can actually interfere with the translation of genes. RNAi is one of those ways where these non-coding RNAs help silence genes. They can target specific genes and block translation, decreasing the amount of protein that's produced. For instance, when I was doing research, we wanted to know if a particular gene, this is SUVAR39H2, right, one of those chromatin modifiers, we wanted to know if that gene played a major role in cell proliferation. Well, to figure that out, you can take the gene out of the process of cell proliferation and see how it impacts the system. If the process slows down or stops, the gene was important. But if nothing changes, the gene wasn't essential. You can do this in two ways. You can either do a permanent removal of that gene, that's called a knockout, or you can do a temporary reduction of its translation or transcription, and that's called a knockdown. siRNA allows researchers to knock down gene expression and see what happens. You can have siRNAs custom made, or if the gene is popular, companies might already have some tested products available. And if you're in the lab and you're thinking about doing siRNA, one of the things that you can do is you can go to companies like Origene, or you can go to companies like Qiagen and find some of these siRNAs, and they'll be available there. Sometimes they'll come in like a premix or come with multiple ones so you can test their effectiveness. What I've noticed is that I like to get siRNAs that have a GFP tag, which is a green fluorescent protein. And that means whenever it's actually inside of your cell, it'll light up and you can see how many are actually affected. So the timeline for this is you plate the cells, you wait till they grow up to about 80% confluency, which means that 80% of the plate is full, and then you do your siRNA transfection. And in these growth assays, we were looking to see, once we treat it with VEGF, how much of the growth would be impacted. You always want to check to see if your siRNA has been successful by doing an RT-qPCR, and then you can look at your gene expression to see if it's knocked down appropriately. And you want about 80% knockdown. In this particular case, what we found out is that if you do a knockdown of silver R398 1 and 2, then you get to see an impact on cell proliferation. So these small non-coding RNAs are all under 200 nucleotides. They're the microRNAs, which are the smallest. They're around 22 nucleotides. And then there's some larger ones like SN RNAs that are around 150 nucleotides. Those siRNAs are involved in RNA splicing. And I don't know if you remember RNA splicing, but RNA splicing is one of the 3 modifications RNA undergoes before it becomes mature and leaves the nucleus. siRNAs, in that particular case, bind to proteins and form these riboproteins called SNRPs. And these SNRPs will then help in cutting out the introns and then joining those exons together. So one major characteristic of RNA is that it's single-stranded. RNAi, or RNA interference, works by recognizing unusual double-stranded RNA and inactivating it. Here's how it works. First, Dicer sees a longer double-stranded non-coding RNA and cuts it into smaller siRNAs. Then that siRNA binds to RISC proteins and forming that RISC complex. One strand of the siRNA is degraded, and the other strand is used as a guide to find the target gene. Once it finds its complementary sequence, Argonaute proteins will then cleave the target mRNA, degrading it. So that's how the siRNAs work. miRNAs, or microRNAs, work in a very similar fashion. Where siRNAs are created or synthetic, these microRNAs are endogenous. And so the microRNAs start off as primary microRNAs, and they form hairpin structures due to their self-complementary sequence. The enzyme DRUSA removes non-complementary 5' and 3' ends to produce your pre-microRNAs. The pre-microRNA gets exported into the cytoplasm and then immediately cleaved by Dicer. It's cleaved because it's longer and it needs to be, cut. But once it's cleaved, then the mature microRNA associates with RISC to target complementary sequences, very similar to what we saw in siRNA. Now let's talk about long non-coding RNAs, or LNC RNAs. So here's what you need to know. One, they do not have start and stop codons. They don't need them because they're not making protein. They can influence chromatin modification, often serving as guiding proteins to where they need to go in order to add the methylation for other proteins. They can, in that way, because they're adding this whole, methylation and are involved in chromatin modification, they can alter patterns of gene expression. So it is interesting that these long non-coding RNAs are involved in chromatin modification. However, they're not directly involved like the DNA methyltransferases or the histone methyltransferases, which actually add methyl groups to those particular spots. However, they are involved in recruiting DNA methyltransferases, and through that, they play a role in cell proliferation, they play a role in cancer, and they play a role in other biological processes. They can also act as competing endogenous RNAs, soaking up microRNAs due to complementary binding sites. All right, so we've discussed non-coding RNAs. Now let's discuss another factor that's involved in gene regulation. This is fascinating too, because this is called mRNA localization. Typically, DNA is transcribed in the nucleus, and then once RNA is processed, it's translocated to the cytoplasm. There, the ribosomes translate it into protein. But sometimes the transcript needs to be translated exactly where the protein will be used. So, mRNAs are directed to those specific areas. This happens via a zip code on the mRNA. And this zip code is a cis regulatory element. Remember, we talked about this, I had a discussion about this in the very beginning of the class. This cis regulatory element tells the mRNA where to go. So let's look at actin mRNA as an example. After it's made and the zip code is added, the zip code binding protein will then bind to that zip code, and the RNA and the zip code binding protein will be exported into the cytoplasm. The zip code binding protein will then bind to a motor protein, which walks along the microtubules carrying the mRNA like cargo. Once it reaches its destination, the SRC will act as a kinase and phosphorylate ZPB1 causing it to dissociate from mRNA. Then the mRNA is translated locally, helping to provide the cell with the actin it needs for movement. In that last slide, remember the zip code binding protein stayed attached to the mRNA until it was phosphorylated. What is phosphorylation? And remember I said the SRC kinase, right? So just remember that and you'll keep that in mind as I go through the slide. So phosphorylation is an example of post-translational modification, meaning that even after a protein is made, it can still be modified. This process of phosphorylation is driven by two types of enzymes. One, kinases. Kinases add phosphate groups. So in that last example, SRC acted as a kinase because it added a phosphate to the zip code binding protein. So kinases add phosphate groups. Phosphatases remove phosphate groups and also alter how a protein will behave or interact. To remember this, I always say kinases kindly give phosphates. So the K, remember? So kinases kindly give phosphates. So, and phosphatases tastefully takes them away. That's how you can remember this. Kinases kindly give phosphates, and phosphatases tastefully takes them away. So while phosphorylation alters protein properties, some modifications determine the protein's fate. Ubiquitin is involved in protein degradation. What do you mean? I mean that if I'm a protein, then the last thing I want to hear, if I could hear the protein, is ubiquitin or ubiquination, right? Because it means that I'm about to be degraded or cut up and no longer functional. And so ubiquitin is involved in protein degradation. This is a major regulatory step because once a protein is tagged with ubiquitin, it's marked to be destroyed. This ubiquination, this major regulatory step, is regulated by ubiquitin ligases. Ubiquitin ligases have the ability to recognize, bind, and catalyze the addition of a ubiquitin residues, which means that it can see the protein that's targeted for degradation, bind to it, and then add the ubiquitin tags on the protein, which will signal that this protein is to be degraded. Here's how it works. So the ubiquitin ligase will recognize the substrate or the protein that is to be destroyed. The ubiquitin ligase will add ubiquitin residues to that substrate protein. The proteasome recognizes this tagged protein. Once it does, it binds to it, removes the tags, unfolds the proteins, and then cuts the protein up into small pieces. That's the degradation part. Once this happens, that protein is no longer functional. Protein's done. That's why I said if I'm a protein, the last thing I want to talk about is ubiquitination, right? But ubiquitination is also the last thing that we're going to talk about. So thank you so much for listening to this lecture. Hopefully you've enjoyed it. Everything that we've discussed today plays a role in regulating gene expression, from silencing to localizing mRNA to modifying proteins that are made. These regulatory mechanisms help us understand how genes are controlled beyond just transcription. So hopefully you learned something today. Thank you for watching. Have a phenomenal day.